Warm greetings. Welcome you all in CEC-UGC Gurukul live lecture. Learners, in previous lecture series of nucleic acids, we had discussed about the biopolymers, and we have learned there biopolymers are the polymers which are produced by living organisms like proteins, all the carbohydrates, sugar, starch, gelatin and nucleic acids. They all are biopolymers because they involved in many biochemical reactions which are very important for our life. Or we can say the plants or all the living organisms synthesized biopolymers. Biopolymers accounts for greater part of the human body, also as well as biospheres. DNA exists on the earth more than 1 billion of years. So today in this lecture we will discuss about the chemistry of DNA and chemistry of RNA. DNA and RNA, the nucleic acids, are polynucleotides and they are family of biopolymers. And learners, DNA is the complex and macrobiopolymers, and it is located into the nuclei of cell, and the elemental composition of nucleic acid is carbon, hydrogen, oxygen, nitrogen and phosphorus. So these are the some elements which makes nucleic acids elemental compositions. In 1869, Friedrich Mises discovered the nucleic acid. Here this is the structure of RNA and DNA. You can see on your screen these two are nucleic acid, one is the RNA and other is the DNA. So RNA is a ribonucleic acid. Here sugar is a ribose sugar, while this is the structure of DNA, and here in DNA the sugar is deoxyribose sugars. So you can see the DNA or nucleic acid is a long, linear and macromolecules, and they are required for the continuity of life. Learners, why do we usually looks like our parents? Because the DNA, this is the DNA, because of this DNA it makes our appearance, who we are, how we looks. So this is only due to of these nucleic acids, RNA and DNA. So it is the most complex structure, and in this structure you can see there are different types of bases which is present in the DNA. Also there is a sugar which is present in the both DNA and RNA. So this nucleic acid carries genetic informations and chemical basis of heredity, and this DNA organized into genes, and also the DNA or nucleic acid clarify the characteristics of genes. Here another characteristic of that DNA is and RNA is, here nucleic acid, which are the most important biological macromolecules as we have discussed in the previous slides. And learners, it is abundance in all living organism, all living things, and the main function of the nucleic acid is encoding, transmitting and expressing genetic informations. And as I told you, the nucleic acids are the biopolymers, they are the macromolecules of biopolymers composed of the long chain of nucleic acid. There is a structure we can see here on your screen. It is a long chain which is identically similar or monomer building block, it is called macromolecules. So, function of encoding is transmitting and expressing it is a composed of long chain of identical nearly monomer building block called nucleotides, and this is another function over here. So, function as I told you in the previous slide, function is encoding, transmitting and expressing all the genetic information, and nucleotides, which is composed of three, especially three units. One is ribose sugar, another is bases, and the third part is phosphate. So, learner, it is very important, DNA encodes an organism's entire hereditary information and controls all the growth and division of cells. As you are looking on screen, so it is a very complex structure of DNA, and it is composed of long chain monomer units. How this DNA is complex? What makes DNA complex, learners? Because you can see on your screen there is a two strand. It is composed of two strands and the structure is helicalized structures. So, these two strands of nucleotides joined together by, there is a base and the base joined together by hydrogen bonding, while one nucleotide in a chain, in a polymer chain, joined together with another nucleotide through phosphodiester bond. So, phosphate group of one nucleotide attack with another nucleotide at third carbon atom, it make a phosphodiester bond. So, in a long chain there is a dehydration, hydrogen synthesis takes place and it's make a double helical structure and make a DNA stronger and stable molecules. In most organisms, the genetic information stored in DNA is transcripted into RNA, and from that RNA the information translated for the synthesis of protein, and the protein synthesis is needed for the cellular structure and function. So, learner, because all the genetic informations which passes from the parents to their offsprings or to their children is only due to nucleic acid present in our biological system, and the protein is responsible for our integrity of our whole structure, like our hair, which is made up of a keratin, our skin, there is a collagen which is also a protein, and also different types of enzyme which are very important for our biological processes or biochemical reactions for survival of our life. So, DNA is responsible for all the genetic information which present in the chromosomes. So, also it is very important function of the DNA is its flow the biological information. How DNA flows the biological information? DNA to RNA, and all the transcripted information passes to RNA, then RNA directed to the formation of protein synthesis, and that protein synthesis is required for the all cell structure and for all chemical functions. Now, on your screen you are looking Friedrich Meiser. He was a Swiss doctor or a Swiss biologist, and in 1869 he isolated the DNA from the pus, white cells of the pus. So, he extracted crude DNA from the pus, white cells of the pus. Although that time his work was not recognized. So, for billions of years DNA has been primary carrier of genetic information, but it took more than 80 years of its importance to be fully realized, and even today more than 150 years after the discovery of DNA, existing research and technology continue to offer more insight and a better answer to the question, why is DNA important? On February 28, Francis Crick and James Watson, 1953, both were the Cambridge scientists in the Cambridge University, described the double helical structure of DNA. That is a molecule which contains human genes. So, human genes contain DNA, and the double helical structure of that DNA is discovered in 1953 by these two scientists, Francis Crick and James Watson. Rosalind Franklin, she was an English chemist and X-ray crystallographer. Her X-ray studies showed that DNA was central to understanding the molecular structure of RNA and DNA, because her lab produced the photograph that helped the universal mystery of DNA. Although she died in 1958 without knowing the significance of her work, and also her work had played very crucial role in contribution for the determining of the structure of DNA. Biographer Brand Maddox called her Dark Lady of DNA. Learners, Watson and Crick shared the 1962, they both shared the Nobel Prize in Medicine with Maurice Wilkinson for determining the double helical structure of DNA. And McCarthy in 1944, in a simple experiment, they have shown the DNA transforming information which carries a genetic information. So, they isolated that DNA from a single strand of bacteria. So, this is the structure of a nucleotide, which is a monomer unit of nucleic acid. And on your screen you can see there is a three part. One is a sugar part, other is a phosphate part, and third one, which is in blue color, it is a base part. As I told you in the previous lecture, the nucleotide is having base. There are two types of bases. One is pyrimidine base and another is a purine base. So, the purine base, which all the bases attached to the anomeric carbon of pentose sugar, and that sugar can be ribose sugar or deoxyribose sugars. So, there is only number 2 position. H and OH can be different on number 2 position. So, on presence of H and OH, the sugar can be ribose or deoxyribose. So, nucleotide had 3 part: phosphate part, sugar part and nitrogen part. And learner, this phosphate part is very important for the formation of nucleic acid, because that phosphate part joined with another nucleotide by the reaction of OH group, which is present on the third carbon atom. In coming slides, we will discuss how the nucleophilic substitution reaction takes place with OH and phosphate group. So, these are the two pentose sugars. So, you have always remember pentose sugar, if at 3 carbon atom of pentose sugar H is present, it is called ribose sugar, and if that H is replaced by OH on 2 carbon atom, it is the deoxyribose sugar. So, on the basis of sugar, we can give them name RNA ribose nucleic acid and DNA deoxyribose nucleic acids. So, these are the bases. These are the 5 bases you can see on a screen. So, what is the difference between DNA and RNA? Because both contain bases. purine as well as pyrimidine. So these bases are adenine bases, which is represented by A, capital A, guanine basis, which is represented by G, similarly cytosine basis, it is C, thymine basis, which is represented for capital T, and uracil, which is represented for U. So the difference between DNA and RNA is only in the that thymine and uracil. In DNA, thymine purine base is present, while in RNA uracil base is present. So always remember, in RNA the purine base is uracil, while in DNA the purine base is thymine. So RNA nucleotides, which is here having uracil, you can see on the screen. So the difference between the structure of that basis is, you can see on your screen, at one carbon atom, U1, 2, 3, 4, 5 carbon atom, there is a CH3 group. So uracil and thymine both are same, both are attached to anomeric carbon of sugar, that is C1 carbon of sugar. So nitrogen and sugar carbon make a glycosidic bond, and the difference is only the presence of CH3 group at 4 carbon atom, 3 carbon atom. This is the only difference, and one difference is that RNA is a single strand, present in the single strand, while DNA is a double helical structure. It is a more stable structure, it is a most complex structure. So the components of nucleic acids in DNA and RNA are made by joining nucleotide in a repetitive way. So in a repetitive way, they nucleotide joined together to form a long polymers or macropolymers or macrobiopolymers. So nucleotides, which is having phosphate plus pentose sugar plus base plus base can be that purine base or pyrimidine base. So nucleic acid is, we can describe pentose sugar plus base and composed of long strand of nucleotide linked by phosphodiester bond. So it is the very important bond. Learner, one question you can ask here, what is the role of sugar in nucleoside or nucleic acid or nucleotides? Learners, the sugar, that is a pentose sugar, anchors the phosphate group, and that sugars linked with phosphate bond make phosphodiester bond. So from that phosphodiester bond, one monomer unit of nucleotide is joined with another nucleotide unit. So you can see on your screen the fashion in which nucleotide is joined. So it is a dinucleotide which contain, if there is a dinucleotide which contain 2 nucleotides subunit, but if there is a oligonucleotides contain 3 to 10 nucleotides unit. So DNA and RNA are oligonucleotides. So in the structure you can see here, there is a 5 carbon atom. At 5 carbon atom there is triphosphate group, and at other end you can see one end having phosphate, which is represented by P, and other end having OH. OH is linked to the 3 carbon atoms. So these two ends, the nucleotide joined together with a phosphodiester bond in such a fashion that one end, which is a 5 carbon atom phosphate group, is attached, while at other carbon atom, that is the 3 carbon OH group, is attached, right. So this is the again nucleotide, and this in nucleotide you can see on your screen there is a 3p phosphate group. So we called it triphosphate, nucleotide triphosphate, and these are the starting material for biosynthesis of nucleic acid. So here the only difference is the ribose and deoxyribose sugar. As I told you in the previous part, if there is a OH group, the sugar is ribose. If there is a H, the sugar will be deoxyribose. So, DNA is synthesized by enzyme which is called DNA polymerase, and RNA is synthesized by enzyme called RNA polymerase, and the nucleotide strand is formed as a result of nucleophilic attack by 3 OH group of one nucleotide triphosphate on alpha phosphorus of another nucleotide triphosphate. So, by this way they can form a phosphodiester bond and linked one nucleotide to another nucleotide. So, when on your screen you can see the OH group, you can clearly see here the OH group in a nucleotide or ribose sugar is present at the 3 carbon atom. So, nucleophilic attacks takes place on the phosphorus group which is P double bond O of another nucleotide, and as a result breaking of phosphorus anhydride bond, elimination of pyrophosphate groups takes place and 2 nucleotides are added at the 3 end, and strand it grow in 5 to 3 direction by attacking from 3 to 5. So, this is a way how one nucleotide is linked to other nucleotide and make DNA a stable molecule, very strong molecules. So, after elimination of pyrophosphate, it is subsequently hydrolyzed, right. In the previous slide we had discussed how the nucleophilic substitution reaction takes place. So, the pyrophosphate is subsequently hydrolyzed, which makes reaction irreversible, and DNA strand biosynthesis occurs in 5 to 3 directions. So, RNA strand is also biosynthesized in similar way using ribonucleotide instead of deoxyribonucleotides. So, there is only this difference, ribose sugar difference. So, like proteins, nucleic acids have both the primary and secondary structure. Primary structure of nucleic acid is a sequence of bases in the strand, and this is the primary structure of nucleic acid, and in this primary structure 4 different kind of nucleotide in nucleic acids is joined to phosphodiester bond, and it can be represented by 5 DG, DT, DC. So, these D, G, DT and DC, it represented the base. G is guanosine, T is thymine and C is cytosine. So, in abbreviated form we can represent this nucleotide strand, nucleic acid strand as DGTAC. So, in this part we had discussed about the what are nucleic acid, what is DNA, and how DNA is very important for our life process. How do we looks like our parents? What is the reason? As I told you, all the genetic information that passes from our parents, from parents to their children, only nucleic acid is responsible, and DNA clarify all the characteristics of genes. In the second part we will discuss some important characteristics and structure of DNA and what is DNA replication. Thank you.